So our keynote speaker is giving the Lepsky lecture, and it's really quite an honor to introduce Dr. Stephen Kingsmore. He's the president and CEO of Rady Children's Institute for Genomic Medicine, where he leads an interdisciplinary team of scientists, physicians, and researchers who are pioneering the use of whole genome sequencing to enable precise diagnoses for critically ill infants. His team was also one of is the Guinness World Record holder for going from a genome to a diagnosis, and doing that just under 20 hours. Before he was there, he worked in genomic medicine at the University of Missouri in Kansas City. His degrees prior to that are from Queen's University of Belfast. He trained in clinical immunology in Northern Ireland and did a residency in internal medicine and fellowship in rheumatology at Duke University Medical Center. So it's really a privilege to introduce Dr. Stephen Kingsmore.
Thank you very much. Can you all hear me okay?
Yes.
Good. So that's Jim Lepsky. This is the Lepsky lecture, so I thought we should get a picture of Jim up there. Jim is a very well-known human geneticist, and it's quite an honor to give a lecture named after him. The title is pretty self-explanatory. These are my disclosures, so I receive funding for some of the work I'll show you from a variety of biotechnology and pharmaceutical companies. The pharmas are basically focused on rare disease drug development, so highly synergistic with the work that we do. I'm going to cover three different aspects of genome science today. The first related to rapid diagnostic genome sequencing, for which I guess I'm best known, and we'll show you some of the recent developments in that field. And secondly, we'll shift the focus from diagnosis to outcomes and explain why that's important and what the ramifications of that are. And then lastly, we'll move from diagnostic genomes to screening genomes. Same population, babies, but a very different focus and remit. And this is something at a much earlier stage of development. So I love to start with a case because it kind of is foundational. It lays the construct, it frames the talk, and it dictates the gold standard. If you're like me and you have a lot of money in your retirement account and it just rapidly decreased, you are somewhat fixated on the concept of a gold standard, since the stock market's not it this week. So what is the gold standard that we all strive for? And I think that is shifting dramatically. I would suggest that the gold standard is to have for any child with a severe childhood genetic disease a period from symptom onset to initiation of effective therapy of approximately a couple of days. That's exemplified in this case. Let's take a quick look at it together. This is a little boy. He's about 6 weeks old when he appears at the emergency department with very early symptoms of encephalopathy. For those of you not familiar with that word, it means his brain isn't working in a generic fashion. And in particular, he has irritability. He's brought to the emergency department. The evidence of encephalopathy earns him a STAT, or emergency head contrasted tomography scan, which shows multiple fluffy lesions in his brain. So he meets the criteria for encephalopathy on CT scan. He's admitted to the intensive care unit. The following morning, we find out two really important features that were not evident in the emergency department. Number one, his older sister, not sure where that came from, his older sister died of something identical a decade earlier, so presented at the same age with encephalopathy, which rapidly progressed to seizures, and she died in infancy before her first birthday without a diagnosis. This was 10 years earlier. This case is now 3 years old, so it was before the era of genome-based diagnosis. Also, we find out that these are first-generation US citizens. They're from the Middle East, and they're first cousins, which raises the idea that indeed this possibly, is a recessive genetic condition. So we then run through the series of steps that are necessary to move from the idea of doing a genome to actually putting that into practice. A couple of steps of gates to navigate, and we wind up with samples in the Genome Center late that day. 13 and a half hours later, we have a diagnosis. Indeed, he does have a genetic disorder. It is recessive, and thankfully for him, it's something which is eminently treatable with high doses of oral biotin and thiamine. So something as profound as a fatal cause of epileptic encephalopathy treated with 2 vitamin supplements. And that started a couple of hours after order, and within 6 hours his symptoms have resolved and he has an excellent prognosis. So this is the era that we are all excited about. This is the new gold standard. Not many kids get the new gold standard yet, but we're going to look at that in some detail today. So that sets the framework for my talk. So for me, this all started with this publication in 2012. In yellow, our colleagues at Illumina in Cambridge, and I highlighted them because who knows, some of them might have been here, and if I hadn't highlighted them, I would have been in trouble. And so this was a collaboration between our group at that time in Kansas City and Illumina here in Cambridge. Most of what we did that was novel, other than the sequencing instrument, was software-based, and that dated back to 2005 when I was in a different job at what was, I guess, a precursor of EBI, the National Center for Genome Resources in Santa Fe, New Mexico, where we built a variety of software systems which ultimately had applications in human genetics and ultimately in diagnostics and in solving cases like this. And so at the time, this was a landmark, the idea that it was possible to decode a human genome, interpret it, and return a result in time to make a difference in sick babies was something that had never really been conceived before. We now understand, many years later, that this was not an isolated event, that in fact we all had been looking at an incredibly large iceberg with 99% of it under the water, and that our conception of childhood onset genetic disease was really quite primitive, and that in fact these are a leading reason for admission to intensive care units in babies, and that they're associated with the highest healthcare expenditures and the longest length of stay in intensive care units, as shown on this slide. We still haven't fully tapped into that. We're still exploring the underwater aspects of that iceberg. We just know it gets bigger every year. So since that original study in 2012 until the time I stopped counting, which was 15 months ago, 44 studies had been published looking at whether a genome was a good idea in critically ill babies. And the result in all 44 studies was yes. And if we take a weighted average of the key findings of all of those studies, it is that just over 1 in 3 babies were diagnosed as having a genetic condition. Just over 1 in 4 had a consequent change in management, and just under 1 in 5 had a change in outcome. Now, those numbers might not be staggering to you, but if you're a neonatologist and I presented that to you with a new strategy, whether it be diagnostic or therapeutic, you would burst out laughing. The idea is ludicrous. Just to put this in context, the next best test for diagnosis, the one which is most widely used around the world, is the microarray, and it has a 7% rate of diagnosis. Consequent to a microarray result, you have maybe a 1% change in management, and it's unlikely you would have any change in outcome ever. That puts it somewhat in context in terms of the transformative nature of a genome in a critically ill baby. Because of the slide I just showed you, the healthcare utilization that occurs in babies with genetic diseases, it was not surprising that this test is net cost savings. So again, in the panoply of new advanced technologies which are brought to bear in modern healthcare, we always have an upper boundary of what we think the system can afford in terms of adopting this at scale. And usually the cutoff is somewhere around $50,000 per quality adjusted life saved. In fact, this test, on average, against a weighted average, saves $14,000 per test ordered. There is no comparator to that. There is nothing even remotely like that in terms of a new technology in the last 2 decades. These numbers are an underestimate because they are all limited to, it's only 8 studies, and they're limited to the initial hospitalization. Those benefits, those healthcare benefits and economic savings continue throughout life. Most recently, it's not published yet, but most recently we modeled it through the end of infancy, so through 1 year of age, and it now stands at $160,000 per child tested. That's pretty good. That was not enough to actually see this be adopted in general in the US. What it took was a move from research studies with parental consent to implementation science studies. This is a newer field, and it's one that traditionally diagnostics never really entertained. You did such studies for novel, maybe social sciences interventions, but the idea of doing this with a diagnostic test was sort of a novel idea, and the whole framework for doing this didn't seem terribly well established when this was funded, believe it or not, by politicians in California about 5 years ago. So they said, we don't want you to do research on babies, we're not paying for that. Instead, do an implementation study. We don't need consent for this. This is being examined as a new gold standard for care. We want you to test Medicaid babies. In the US, in intensive care units, about 1 in 2 babies will have their bill paid by Medicaid. So it's a government health insurance provided for low-income families. So that was the test population, and we did this all across our state, and there's a lot of diversity within our state in terms of cities and countryside, but these were low-income families. We recapitulated the diagnostic proportion, so here it was 43%, and the clinical utility, the change in management, 31%. And we also recapitulated the economic findings, $3.7 million across 184 babies tested. So in response to that, we had a dramatic series of events over the last few years, which was that state after state started to announce a policy of coverage for rapid diagnostic genome sequencing in the Medicaid population. This is the map as of last month, March 2025. We have 18 states who now have policies, coverage policies, for a diagnostic genome for a baby in an intensive care unit. Tragically, to my mind, only 2, 4, 6, 7 of those states have a carve-out payment, so in the rest of the states, this is part of the package which is given for each admission. So each admission gets reimbursed at a certain rate, depending on what we call the DRG, the diagnosis-related group. And in those other states, there is no markup on that for ordering a genome. So in those states, there's really no incentive for ordering a high-cost test other than that it is likely to benefit the patient population. So as you can see, most of the states does not yet have these policies, and in many of the states, these policies happened in the last year, which means that they haven't really trickled through to individual hospitals and individual accounting practices, and so we as yet do not have much in the way of national adoption. This is my best estimate of it currently. It's down here. It's fairly depressing for something that's now 13 years old, and it has all of those published studies. About 4% of the babies who might benefit are likely to receive a rapid diagnostic genome sequence in the US this year. And there are 3 different groups who are not receiving this. First of all, many neonatal intensive care units aren't using this at all. But in those who are using it, they tend to underutilize it. So NICUs who do order rapid genomes just under-order it. Second of all, as I said, most NICUs have never ordered a genome sequence on any baby. That's fairly tragic. And then lastly, we now realize, based on very recent data, that the policies that exist don't really cover all of the babies who have needs. And so the actual policy coverage information needs to be revised. So when you add all of that together, we have a lot of work to do. And it's a different type of work than what I grew up doing. So a couple of things. First of all, what are these policies? So one of the complicated things about rare genetic diseases, and you know this, there are over 10,000 of them, is that they affect every organ system in the human body, and they're very diverse. And so no single simple catchphrase is sufficient to identify babies who might benefit. Instead, you go organ system by organ system, presentation group by presentation group. This makes it incredibly difficult for a neonatologist to know whether an individual child will benefit or be covered for this test or not. They make sense. I mean, these are great reasons, and for each of these I could rhyme off cases and lives that we've saved. The one I just showed you, that baby was starting to have refractory seizures, had a family history of something that might have been a genetic disease. So you can see where different babies will fit in. But it's very complicated. So in response to all of these factors, we're now moving on into bits that are not really genome science, they're more human science. How do you take a test modality, a genome, and actually have that become that gold standard? How do you get to 100% of babies in the US and around the world actually receive this benefit? And this turns out to do an awful lot with physicians and where they're at in their heads. So for them, they have two areas of deep need. But let me just frame this first of all. So this represents the progression of a typical genetic disease in a baby. They progress incredibly rapidly. Basically, the older you get, the slower your disease will progress, as a rule of thumb. And so in newborns, they evolve in front of your very eyes in terms of disease severity. That's why we need speed. People my age, we just don't get sick that quickly. So hitherto, our focus as a community has been on TTDX, time to diagnosis. We have been focused on this, as you said in your introduction. We have 2 Guinness World Records for fastest genome. And so we do this well. We are really very good at going from a test order and receipt of a blood specimen to return of a report. We can routinely do this in a couple of days. What we really need, though, the gold standard that I introduced, is not time to diagnosis. It's a surrogate. It's a biomarker of what the baby really needs, which is time to treatment, time to optimal treatment. From symptom onset, in this case, the proxy is NICU admission, to precision intervention delivery. And so there are 2 areas of need. The first, on the front end, how do we get the babies who will benefit to be identified and the tests ordered as quickly as possible? And that's 2 problems, or maybe 3 problems. First of all, we need to extract from the medical record the phenotypes, the presentation of that child's illness. We desperately need that to do a good job of interpreting the genome. The genome today is not interpretable absent a ton of phenotypic information, and the more phenotypic information, the more broadly and more precisely we can interpret what we see in the genome. So we need to manage through that. Doctors are not used to that. They're used to ordering stat CT. That's it. They're not used to pages and pages of phenotypes or their quantitative nature. Second of all, I already talked to you about the difficulty in identifying which babies do and do not meet criteria in your hospital, and then the practicalities of obtaining consent. And in some places you need both mum and dad, and in some it's mum or dad, and in some cases it's a guardian. This becomes complicated. In some cases you need trio samples, mum, dad, and baby. In other cases, baby alone. So this can take weeks. So how do we solve for this? And then second of all, how do we go from the fact that we're delivering what I might call gibberish, so unintelligible information in a foreign language, to a highly trained specialist, a neonatologist? They didn't learn how to read genome. They don't know how to read genome. Furthermore, our reports are couched in all kinds of arcane specialty language that make a ton of sense to a medical geneticist and no sense to anybody else in the universe. For example, we don't call something a mutation. That's a term that we might understand. No, we call them variants. We call things pathogenic and likely pathogenic and uncertain significance. And so time and time again, we return a result to a neonatologist and we get an immediate phone call and they say, did you make a diagnosis? And we're like, yeah. Did you read the report? Yes, I read the report. Did you make a diagnosis? And you then walk them through the gibberish and put it in regular English. Furthermore, and this infuriates me, our field gets more and more and more complicated in terms of its notions. We now no longer actually report the disease we think it has. We will say, your child has an SCN2A associated condition. Now, the neonatologist has no idea that SCN2A is a gene and no understanding of what SCN2A stands for, and there's no way for them to easily look that up. That's not in a medical textbook, and so we've lost them, right? And so they'll then say, what disease did you diagnose my child with? And then the response will be, we're not sure, but we know it's in this gene. You understand the problem that we have? It's like two people born in a different system in a different era trying to communicate with no interpreter. So we need that, because what this baby needs is actually not a label, they need a treatment regimen. It gets much worse. Let's keep going. So it turns out when you actually try to do this, and this is a system we've built that tries to do this, it tries to solve for that. And so we think we've gotten all of the information items there in a single place, so that instead of returning an unintelligible report, we can believe that that baby will get the treatment that they ought to get, like biotin and thiamine, delivered orally 4 times a day via a nasogastric tube in these doses. But whenever you go to the primary literature and try to identify this, you find it spread all over the place. For most of these diseases, there are not what we call clinical practice guidelines. Nobody has formulated the gold standard. And when you look through it, you find only that 16% of the treatments which are advanced for these therapies actually have efficacy, actually have evidence of efficacy. We're all becoming aware of this now with these mysterious new treatments like vitamin D for measles, right? Rare diseases in general are like vitamin D for measles. There is not a consensus or a clinical practice guideline in terms of what is the acceptable treatment for that condition. Furthermore, it keeps changing. People keep publishing manuscripts, and each year we need to re-review the literature. So this now exists. It's not yet really in widespread use, and it's not been pressure tested. So we need to do an awful lot more work to see, is this actually solving the problem it's supposed to solve, and will it change outcomes? Okay, I'm wrapping up the first part of my talk, and so this is sort of the summary statement. If you have a baby, why should that baby get a genome if they're in an intensive care unit? And these are not the lowest level of neonatal intensive care. These are the higher level, what we might call regional neonatal intensive care units. Number one, you'll change the bottom line of your hospital. That's really important these days. Number two, you'll shorten the length of stay. That's really important because in general, there are more babies who need a NICU bed than NICU beds available. Number three, I hope I've shown you, it's the new gold standard. Number 4, it's the gateway to getting them that therapy. And those therapies are often surgeries. Again, we always think in terms of pills and FDA-regulated medications. It's often a dietary intervention. It's often a surgical intervention. Our focus is improving outcomes. Another area that's really important for many of these diseases is the difficult decision to move to palliative care. So what if this child is going to die and there isn't an effective treatment? At what point do we think through how to start to withdraw care? This is a very delicate decision. It's one where every family has to make up their own mind. What's right for one family is not right for another. It has many influences, whether those are religious or social or familial. The fact that you have a diagnosis, the fact that you have a prognosis, gives the parents back control. It means that doctors are no longer making decisions on behalf of a family because you now know the answer. You can give them back control. They can make the choice. I would like my baby to come home for whatever period I have left. No, I would like them to stay in an intensive care unit because I'm hoping beyond hope that this will be the mildest case ever and what you just told me won't be true. This is, believe it or not, the area where we find parents are so happy. It's completely paradoxical to me, but this is the single part where time and time again parents will say, thank you so much. And then lastly, if we don't diagnose a baby in the intensive care unit these days, that probably means they won't get a diagnosis, or at least they won't get a diagnosis in the next 5 years, because overwhelmingly families are confronted with this phenomenon that we hope to make extinct called the diagnostic odyssey. Okay, switching gears. I want to talk about new technology. So this all started with Illumina's rapid run mode on the HiSeq. And about a year ago, we got a call from them to say, hey, we got some cool new stuff. Do you want to try it out? And we said the same as we always say, which is, you bet. So we started sending them samples of babies who subsequently we diagnosed, but who you couldn't tell that based on a conventional Illumina genome. And they were able to sort them out. So here's how it works. It's really brilliant and simple. Instead of making a genome as a chemistry experiment with lots and lots of tubes and robots and stuff, we're going to make the genome inside the flow cell. So we're going to take single-stranded, high molecular weight genomic DNA and inject it into the flow cell where it will sit down. As we all know, these flow cells have primers in them, and it will anneal, the single-stranded DNA will anneal to the primers, and here it's high molecular weight, so it will anneal to a series of contiguous nanowells. So then you cleave the DNA in the flow cell, make the library the same way as usual with this PCR technique, and sequence it as per usual. So these bits are identical, other than that this is performed in the flow cell. And the result is that you get a series of paired reads, but the 5' and 3' to the index read is going to be in an adjacent well. So now, based on the two-dimensional geography of the flow cell, you can actually assemble a genome. Now that sounds like a nice idea. Does it work? Yeah, it really works. About 70% of the time, the next well has the immediate 5' or 3' sequence. 70% of the time. So you can do the math. Each time you jump down, it's 70% of 70% of 70% of 70%. We're doing 30x genome on average. And so the math solves itself. And in general, we have nano-assembled genomes. So I got so excited about this that I decided I was going to rerun my genome. I haven't run my genome in a decade. Back in the early days, we were always looking for volunteers, and so I would give some blood. And so I had my genome decoded lots and lots of times, and I thought it was time to do it again. Why the heck not? So they let me come into Illumina and do it. I'm 64. I haven't made a library for genome sequencing since 2008. Right, so I'm a little bit rusty, and I don't have the coordination that I used to have, and I sure have difficulty seeing up close. And there I am. You can see I'm kind of focused. Oh, I look kind of focused. So it only took 10 minutes. Literally, your CEO can do it. That's awesome. What that translates into is a regular hospital laboratory can realistically set up a genome. If they can do ELISAs, they can do genomes. If they can do fingerstick glucose monitoring, they can run genomes. But maybe more exciting for you all is this fact that we now have the reality of phasing across genes and genomes, and we now have all the benefits of a long read and then some in terms of the complex, large variant stuff. So let me just, I don't know if any of you have even heard about this. It's called Constellation. How many of you have ever heard of Constellation? Okay, a few of you, maybe a third. Cool. So it's not available until next year. They still have to build out software bits. What it means, so this is a new plot. You haven't seen this plot probably before. We're used to IGV, the Integrated Genomics Viewer. This is a new plot. It's a density plot. And so basically the color map is how dense is the read information. And we're looking here at the observed versus the expected coordinates along a genome. And so if you have reference genome, we're looking for structural variation. If you have a reference genome sequence without structural variants, it'll look like that. A deletion will look like this. You're missing that bit. An insertion will look like this. You've got an insertion. And this is the coolest one. This is what an inversion will look like. We just can't see inversions with short read genomes. It looks like a pair of bow ties. This is our future, folks. So here's what it looks like in reality. So now we're looking at the factor VIII gene. So factor VIII, if you break it, causes hemophilia A. 1 in 5,000 boys will have hemophilia A. They will recurrently bleed into their organs unless they're given hemoconcentrate to replace their factor A activity. Super common disease. And the commonest mutation, pathogenic variant, is this one. It's a 600 kb event, and it is an inversion. So this is the normal sequence of the factor VIII gene region, and it has this repeat sequence in intron 22, 9.6 kb, and there are 2 copies of it in the counterclockwise direction, upstream of the gene, and so this gene tends to recurrently flip through those breakpoints, and it's clean. It's clean to the nucleotide. So there's no signature at the start or end that you can detect this with. And so here's the bow ties. So we sent them a whole bunch of samples, and they got 16 of 16 right. This represents 45% of hemophilia A. Up until now, we just couldn't pick up hemophilia A reliably with a short read genome. Now we can. I don't know about you, but that gets me really excited. We're going to start to see all kinds of new structural variants in regular genomes at scale that we hitherto couldn't. And the beautiful thing about this to me is, I mean, I'm a big fan of long-read genomes, but they come with an awful lot of baggage for a clinical lab in terms of validation, in terms of software selection, in terms of the complexity of your process. The idea that we can get this on our current Illumina boxes is awesome. Okay, switching gears. We're staying with Constellation, but we're talking now about a specific case. This is Connor and his mum, Kelly. Connor is 14. He's wheelchair bound. He's in San Diego, so most of my stories are San Diego. That's where we're based. He was very like that baby I showed you earlier. At a month of age, he started to seize. And this was a long time ago. There were no genomes, so he went undiagnosed. To his seizures were added developmental regression. So he met milestones, and then he fell backwards, and he lost them. So for example, he can't walk. Previously, he used to be able to walk. He can't speak. On top of that, then, he developed autism spectrum disorder. So very concerning behavioral episodes. So he went through the diagnostic odyssey that I mentioned, and about age 8 or 9, he was diagnosed, and we'll take a look at his mutation or pathogenic variant. And this is it. It's right in here. It's an arginine to glutamine at position 853. This is amino acid 853 in the SCN2A gene that I mentioned earlier, the type 2 voltage-gated sodium channel alpha subunit gene, which together with beta 1 and beta 2, this is the alpha, makes up this ion channel, which you find in human brains. And so his mutation has been seen in 11 human beings, so it's recurrent. And it's a mixed gain and loss of function. So we do electrophysiology on these to understand why does it cause seizure disorder. And in this case, it both has aspects of gain and loss of function. And that's probably enough on that slide. And so here's where it is in terms of the synaptic cleft. So both in inhibitory and excitatory synapses. That's tough to say with jet lag. Excitatory, say that 3 times fast. So here is that ion channel. So it's on the presynaptic hillock, and it changes the voltage across that region of the membrane, and that in turn changes the size and the repetitiveness, the length of the depolarization that happens when that axon fires. It's one, as you can see, of many molecules in this region, all of which can cause fulminant seizures. So, these are anti-epileptic drug-resistant seizure disorders. There are over 1,300 genetic causes of epilepsy. And in blue boxes are the agents, the anti-epileptic drugs that target individual proteins. And he had been tried on most of these and they just didn't work. He had refractory seizure disorder. I love this slide. I'm going to show this until I'm 85. Okay, so this is a journey, and one of the fascinating things that Connor's mum realized at some point was that Connor couldn't speak, but that didn't mean that he couldn't communicate. He was thinking in his head, he didn't have a way to get the words out, and so they found that he could use a word board to actually speak, to communicate. And so he was able to start to elaborate with his mum about his disease. Okay, so why this gets super exciting is that we now live in an era when a little boy like Connor can receive a therapy built for him. And that's exactly what happened. They're called antisense oligonucleotides. And this is sort of the architecture of a canonical antisense oligonucleotide. It's got a mixture of different nucleotides. Some are synthetic, some are native. And it's got 2 stabilizing arms and then an RNase H binding centerpiece. And this targets a place in the genome uniquely, sits down on top of the pre-mRNA, so before splicing, and targets that pre-mRNA for RNase H degradation. So you use this on the affected copy, the affected protein copy, to degrade it so it's not expressed, so you still express the normal copy. Got it? Antisense oligonucleotides. Why this is exciting is that there is now a charitable organization called N-Lorem, which was founded, 8 years ago, that makes N of 1 therapies. So they'll take a child like Connor and they'll qualify him to make one of these just for him. And they've navigated with our Food and Drug Administration an approvals process that takes less than a year. So basically, this backbone is now validated as a therapy, and so they don't worry too much about that, and instead they focus on efficacy and safety of the specific features. And they only will do this for very severe childhood diseases and very rare childhood diseases. So he went through this. Normally you can do this in 1 year, if you can believe it. In his case it took 4 years because he was the first patient, and also because his particular variant was kind of tough to target. So this is his variant. It's way down here. It's now we're looking at IGV, Integrated Genomics Viewer. We're now back looking at constellation. So some of you, your jaws ought to go, huh? This is a short read genome, but look at the coverage plot. There are no gaps. We're looking at the entire gene, and we've resolved the 2 haplotypes. This is constellation. Constellation routinely gives you phased genomes. And so what we can see in this case is that here's Connor's variant. It's heterozygous, so it's on this copy, haplotype 1, not on haplotype 2. And what we're actually targeting is the reference genome way up here, 60-some kilobases away. And on the opposite copy, the healthy copy down here, and this is intronic, is a SNP, a single nucleotide polymorphism, benign, common, right? So that's what we made, or what they made for him. It targets reference genome sequence in the pre-mRNA, quite a distance away from the causative variant. So as I said, it took almost 4 years to get this from concept to injection of his first dose. It's given intrathecally, so he needs general anesthetic. And this started, as you can see, in March last year. And I love this. A reporter interviewed them before the first dose and said, "What are you hoping for from this therapy? You know, you've been waiting for it for forever." They were part of the foundation of N. Lorem. And Mum said, obviously, "I just wanted to stop seizing." Connor, and he had to type this, right? As I said, this whole inner life has now started to become aware of it. He had much grander aspirations. I want to be able to walk. I want to be able to talk. Well, it turns out both of them got their wishes true. So he's now had 6 doses, and you start off at a really low dose, and then 3 months later you give a higher dose. So he's still on the step up. But believe it or not, as of the first dose, seizures stopped. So it works. He's had no adverse events. And what's really cool is he's walking again. Now, that rewrites the textbooks. That's not supposed to happen. You're not supposed to be able to get that ability back. These are fixed phenotypes. That's just not the case, folks. And so he's not really steady on his feet, but he's walking independently. Okay, so we got very excited about this. We get very excited about it for 2 reasons. One, this whole N of 1 therapy is exceptionally exciting because most of our kids won't get an effective therapy. But number 2, because this is targeting the reference genome. And so how many more kids might fit this? They need a pathogenic variant in trans with a SNP that's present in 20% of the population. So we started to explore that, and this is in progress. We've tested, this is a common thing. We've tested 7,000 babies with rapid diagnostic genome sequencing, most of whom were having seizures. We have 17 who were positive and consented for research. Of the first 11, 3 are positive. So they've got that common SNP in trans with a disease-causal allele. So potentially, we have several new kids that we can explore for utility of this therapeutic. N of 1, no more. N of 1 to N of few, and maybe N of many, we're going to see. So Connor has developmental epileptic encephalopathy 11, but he also has autism. But some of these patients develop bad schizophrenia. Some of them have other epilepsies. And so we're quite excited about this, and we're moving through this series of steps now to say which of them have gain of function or mixed gain and loss of function diseases, and will any of them respond to this. Now, I've left way too little time for the last bit, so I better hurry up a bit. So we're talking now about something entirely different. Same thing, genomes, same population, babies, but we're talking about screening. And this slide is just supposed to say that, hey, they're very similar, but they're very different. It's a belt and braces approach to solving the problem of how do we get these babies to have optimal outcomes. I don't have time for this story, but this is a little boy, I just tell it really quickly, who tests positive for SCID on his newborn screen. He gets a rapid genome. It turns out to be Artemis SCID, that's the gene. He gets gene therapy. He's cured. He never has his disease. Boom, ex vivo gene therapy. Okay, for this project, this is now future looking. We want to skate towards where, this is an ice skating metaphor. You may not know ice skating. We want to skate towards where the puck will be in about 3 years when this will start to be implemented. So one thing that's very exciting is that many, many new gene therapies are being approved worldwide, and that this is only getting started. This is an exponential curve, and we can anticipate that in the future we will have thousands of genetic therapies. We also know that the cost and the speed of genome sequencing are exponentially decreasing. And so although what I'm talking about today seems like a completely crazy idea, let's screen 40 million babies worldwide with a genome for genetic diseases, these are some of the reasons why it probably will happen. Okay, we've built a platform. It's now in version 2 to do this. It doesn't look like a normal genome sequencing platform because it can't be. This is a very different problem. These are healthy babies. They have no phenotypes to guide our interpretation, and so we need an automated system that self-qualifies every variant. And we need to move away from the idea of, is this a VUS or a pathogenic variant? Those concepts have no meaning in screening. We're doing risk assessment. So we want to attribute a baby with a specific risk for a disease that has an intervention. Completely different paradigm. And so we got to build it from scratch. Okay, how many? I've got 5 minutes left. I think I got 10 minutes for questions. I'm going to steal a wee bit from my 10 minutes for questions, if that's okay. So what are the 4 major things that we need to do to solve this problem? Number 1, we've got to reduce the cost of clinical genome testing by 40-fold. Now, part of that is Moore's Law, it's skating towards the puck, but a lot of that has to do with complete automation. We don't want humans to be engaged. We want a machine to say this child is at risk for that. That's what we want to do. Wow. Brings up ideas of Gattaca, doesn't it? How many of you have seen that movie? Seen Gattaca? Okay, go out and see Gattaca, would you? Goodness sake, you shouldn't be in human genetics. You haven't seen that and like know all the punchlines, because it's kind of like an ethical, legal, and social part and parcel of what we do. Okay, false positive rate is a huge problem. We are used to the fact that a baby is suspected of having a genetic disease, and so we order a genome test. If a baby is not suspected of having a genetic disease, they appear healthy, then the likelihood of them having a pathogenic variant is incredibly low. And what that means is that our variants, which are very carefully distinguished as pathogenic and likely pathogenic, for diagnostic cases are pretty useless when it comes to screening cases. The information is not really useful. And on top of that then, I told you this earlier, we have not had the discipline as a community to say what's the phenotype in general associated with that pathogenic variant. And we want to screen for severe diseases, not mild diseases that don't really need therapy. Then we have the problem that many of these ultra-rare diseases are in fact ultra-rare, which means that when you do a clinical trial, you can't actually prove your test works because you get no positives in it. You get 100,000 true negatives. So that's a bit of a problem. Are we going to do a clinical trial of 1 million babies? That'll be expensive. And then second of all, we have the fact that each genetic ancestry has its own unique set of prevalent conditions and its own unique private set of causative variants. And so a test that works in Northern Irish guys will not work in, I don't know, Ugandans. It just won't. We need to think that through and make sure that we validated this in many ancestries for which we don't actually have good reference populations today. I don't know if you've dug into this again. Must do. Read into the 1K genome set and ask yourself how many human populations did it actually cover and to what depth. That will lead you to a sobering conclusion. And then lastly, we've got this same phenomenon of the workforce doesn't understand this. How are we going to implement this with regular MDs, with regular family physicians? Okay, so 3 core concepts. Number 1 is you've got to deal with diplotypes. You've got to get your mind off alleles and variants. I know we all love them, but they're not much use for screening. You've got to get used to thinking about diplotypes. That's the pair of haplotypes in each gene and all of the associated variants. Second of all, you've got to get used to this idea Up until now, genomic information was largely derived from aggregated populations. Let's gather 500,000 genomes and query them and make major advances in our understanding. Well, unfortunately, most genomes these days are siloed away in subsets, and that won't work. There are over 10 million genomes around the world, and 90% of those are not accessible for an aggregated analysis. So the solution is instead send somebody your code, ask them to run the query once they've inspected the code and are happy with it, and send you back the aggregated results. You'll get the same thing. What you need back are diplotype counts. You never need to be able to inspect their data. Will that work? Third core concept we've been using is this one, which is UK adults and adults in general are wonderful controls for newborn screening. I can't have a very severe childhood onset genetic disease. Why? Well, I'm still alive, duh. So, tremendously powerful in picking up false variants. Okay, I'm sorry about this. This is like the coolest part of the talk. I'm going to rush it. Okay, if you run, let's just say you run a genome-based newborn screen on UK Biobank, which we did a year ago. And here we're looking at cystic fibrosis. It turns out that 2.3% of the population screen positive. That's not very good, because the real incidence in UK adults, and this is biobank-aged adults, so 40 through 70 roughly, is 20 per 100,000, not 2.3%. So 99% too many positives. So you can't run that test, even for cystic fibrosis. However, you can build a mathematical model that I don't have time to explain and come up with a cutoff value for that gene, which when you apply it, so this is the commonest variant to the rarest variant, looking at 54,000 variants. Here we're looking at the cystic fibrosis locus. All the red dots are false positive diplotypes. You're able to solve the problem and by removal of a few variants, get to a solution where you now have the test work in the UK Biobank. So you can scale that up, you can do that for hundreds of diseases, and you can now take that query and run it in a federated manner where you are sending this piece of software to federated data sets worldwide. They are returning a result set that looks like this. It's just diplotype counts, and then you run the math and you're able to take the learnings and solve for each disorder. This actually works. When we did it on UK Biobank, we were able to take our false positive rate from 74% to 2%. Oh man, this is such good data. This is a clinical trial. I'll just show you very, very quickly. We did this last year. So we took babies in our level 4 neonatal intensive care unit who were not going to get a genome, right? No, this baby is not genetic, no genome. And we enrolled those consenting parents and got from their babies 4 tests: a California newborn screen, a diagnostic genome, our new test, we call it Beginnings, that's genome-based newborn screening, and then we also did one, just for chuckles, with 1,400 different disorders. So what did we find? We found that 29% of those babies who weren't supposed to have a genetic disease did have one. Woohoo! I told you the iceberg keeps getting bigger, and that most of those were going to change management. So even in our NICU, in our hospital, we're not doing enough genomes. And then we found that the genome-based newborn screen with those fixes works pretty well. We had no false positives. We had 4.2% true positives. And that compares with the California newborn screen pretty well. And then interestingly, we find that when we take this up to a newborn screen with 1,353 genes, we take it up to 10 or 11%. So 11% true positives and 10% clinical utility. So now we're doing this. We want to do this worldwide. And our goal is to move into implementation, the Project Baby Bear thing I told you about, so that we get policies and to up it to 1,000 diseases. And we're in the process of that. We've set up a consortium to do this. I'd love to have more names on this of people from the UK. Don't have too many from the UK on it yet. Those were my starting points. They're my finishing points. And I got to say thank you to many, many, many people for all of this work. And this means to God for creativity, to friends for help. Thanks very much, and I apologize for rushing the end. We got 6 minutes, so far away.
Beautiful work. Quick question. For SCN2A, haploinsufficiency is the leading cause of autism. And so once you fix this with ASO, what is the phenotype of the patient now? Because you're still left with only one copy of SCN2A now.
Yeah, that's a great question. The bottom line is we have no idea. So this just happened. This technology just got invented. We have an index case, a little boy with autism. I didn't have time to tell you, but on his typing board, we said, how do you feel after you get your medication? And he said, my brain feels calmer. So I don't know about you, but that sounds pretty good for a kid with severe autism, right? That number one, they're able to respond and they're able to, provide that type of response. But it's N of 1. And so until we get broader exposure on this, I don't know. And he's only been on the treatment now for a year, so we're still figuring out what it will mean for him.
Yeah. Question down here? Yeah.
Hi, Fauzan Kurai, Saudi Arabia. Great talk as usual. I just wanted to ask you about your vision for including other modalities to supplement your vision for genomic newborn screening in order to make it even more sensitive and specific in picking up conditions, like combining it with metabolomics, for instance. Is that in the works in your project?
There are others, Fauzan, Michael Gelb in particular, focusing on metabolomics. There's actually quite a large community focusing on metabolomics. That has been the mainstay of newborn screening traditionally, mass spec. And so saying, what can we do more with mass spec, opposed to looking at 38 conditions? And undoubtedly that will help. And undoubtedly having a genome plus a metabolome would allow many of those metabolite signals to get some context. So that's quite exciting. And furthermore, we're now starting to look at some, what we might call polymetabolic signatures. So very similar to polygenic risk scores, but with metabolite peaks. And starting to build biomarkers for things like infant death. And so there's great hope that we can utilize, as you say, more than one modality truly to give a richer profile on newborn testing.
Thank you.
I'll ask a question. So there's other long-read sequencing technologies than Constellation. I'm sure you're asked this often, but what's your view on those and their utility in these environments?
It's a great question. I don't know. It'll take several years to answer that. As I said, the impediment from a clinical perspective to long read today is that the whole software side, the clinical software side, for the downstream analysis and turning that into a reportable event is still in its nascency. And so that is a major problem. We now have generated, as I'm sure many other groups have done, many nanopore genomes in strikeout cases of babies with short read genomes, and then you get stuck. Now what do I do? How do I analyze these in a reproducible, clinically acceptable manner to turn them into a diagnostic report? That's not to say the Constellation has solved that, but at least for Illumina sequencing, we have widely deployed, somewhat generalizable platforms for taking it all the way through a report. And also we have the installed base. So from a practical standpoint, this could be transformative for regular clinical labs who right now are kind of stuck. They cannot, gear up and have 5 bioinformaticians building custom pipelines for them for their own in-house cases.
Yeah. We also have questions online, do we, or no? Okay.
I've got one here too.
Oh, one back there, sorry.
Hi, thank you. It's impressive that you have such a high fraction of these NICU cases who get treatments based on their diagnosis. I was quite surprised it was as high as kind of 30%, because in this deciphering developmental disorder study that I work on, which I'm sure you know about. It's not NICU kids, but other severe disorders. I think it's much lower than that, more like 2% or something. And I'm just wondering, I guess this is to do with the characteristics of the kind of conditions that are presenting to NICU, that they're more likely to be metabolic disorders, or you mentioned that surgery was a common intervention. So do you know why it's so much higher than for the broader developmental disorder population?
Well, without being provocative, could I ask you why yours is so low? What are you working on?
Well, maybe that's just the NHS in the UK, could be.
But what disorders in particular?
Oh, well, look, it's broadly, mostly neurodevelopmental disorders, but we have a lot of these, epileptic encephalopathy cases as well. I'd have to look at the breakdown by the different class. But.
Okay. So in general, things like ketogenic diet, specific anti-epileptic drugs. So it might be partly the definition of what I call a tailored treatment. It's where the genome has informed your decision, let's say, on which anti-epileptic drug you're going to choose. I'm not going to choose a sodium blocker because I think I have a gain of function or a loss of function mutation in that locus. It's things like that where you're taking the guessing game out of it and practicing precision medicine, maybe with conventional therapeutic paradigms in light of a genome. I think that might be where we're being lost in translation.
All right, I think we hit time, right? Yeah. So let's thank Stephen again. Thank you very much. That's a fantastic talk.